Early growth response protein 2 is a protein that in humans is encoded by the Egr2 gene. Egr2, also termed CROX20, is a transcription regulatory factor containing 3 zinc finger DNA binding sites and is highly expressed in a population of migrating neural crest cells. It is later expressed in the neural crest-derived cells of the cranial ganglion. The protein encoded by CROX20 contains 2 cis2 has 2 type zinc fingers. CROX20 gene expression is restricted to the early hindbrain development. It is evolutionarily conserved in vertebrates, humans, mice, chicks, and zebrafish. In addition, the amino acid sequence and most aspects of the embryonic gene pattern is conserved among vertebrates, further implicating its role in hindbrain development. When the CROX20 is deleted in mice, the protein coding ability of the CROX20 gene, including the DNA binding domain of the zinc finger, is diminished. These mice are unable to survive after birth and exhibit major hindbrain defects. These defects include but are not limited to defects in formation of cranial sensory ganglia, partial fusion of the trigeminal nerve, V, with the facial, 7, and auditory, 7, nerves. The proximal nerve roots coming off of these ganglia were disorganized and intertwined among one another as they entered the brainstem, and there was fusion of the glossopharyngeal, 9, nerve complex. The early growth response protein 2 is a transcription factor with 3 tandem C2H2 type zinc fingers. Mutations in this gene are associated with the autosomal dominant Charcot-Marie-Tooth disease, type 1D, Dejerine-Sodda's disease, and congenital hypomyelinating neuropathy. 2 studies have linked Egr2 expression to proliferation of osteoprogenitors and cell lines derived from Ewing sarcoma, which is a highly aggressive bone-associated cancer. New research suggests that CROX20, or the lack of it, is the reason for male baldness.